I'm coming from the same team as Stephen, and today I'm going to be presenting our results on the single cell track of the CAMDA Health Privacy Challenge. We essentially took this as an opportunity to benchmark various state-of-the-art synthetic data generators for single cell RNA expression data. So this talk will be comprised of 4 parts. I'm going to start off with an introduction and some motivation, and then we'll move on to methodological descriptions of various pre-published methods for synthetic data generation of RNA counts data. Following that, we're going to go over the metrics for data quality and for privacy based on the pipeline established for the Health Privacy Challenge, and we'll conclude with further discussion of the model privacy. So to briefly motivate this, as we all know, single cell RNA data is extremely useful to quantify expression at the single cell level, which enables us to determine trends at the cell level, to piece out cell heterogeneity, to determine various cell types and differences in gene expression between them, and so forth. So this has many applications in cancer research, and researching tumor types, etc., and additionally in helping to identify data trends which may be masked in aggregate bulk data. However, there are still some challenges when dealing with single cell RNA-seq data. First and foremost among them, there may be a class imbalance within the data you have. Certain cell types may be rare, certain experimental conditions may be rare, and this can limit the ability to train a ML model on single cell expression data. Furthermore, as we've discussed extensively, there are a number of privacy concerns related to distributing any sort of single cell expression data, or even worse, distributing single cell sequence data. And nowadays, there are increasingly privacy regulations governing the sort of distribution you can do with this single cell expression data, for fear that donors in single cell studies may be de-anonymized. One solution to this is using synthetic data generation algorithms, which allow us to mimic the distribution of actual single cell expression counts without revealing the exact data that was obtained from sequencing. And this can help to alleviate concerns surrounding donor privacy and also to alleviate class imbalances within training data. So to move on to our overview of some existing state-of-the-art methods, we select 4 methods which fall into 2 broad categories. These are statistical methods, which essentially fit distributions over existing single cell expression data and sample from these distributions, and deep learning methods, which involve more complicated neural network-based approaches, in particular diffusion approaches, which have become popular recently. Probably the most sophisticated of these statistical models is scDesign-2, which fits an individual marginal distribution to each gene within a single cell data set. And it has some simple heuristics to determine what a good distribution for a particular gene may be. So for instance, overdispersed data are fitted to a negative binomial distribution, whereas non-overdispersed data are fitted to a Poisson distribution. And depending on the sparsity of the data, you may choose to use zero-inflated versions of these distributions or not. Now, where scDesign-2 really distinguishes itself from other statistical methods is in its use of a Gaussian copula. This is a dependency structure which allows you to compute a joint distribution over multiple independent marginal distributions, even if no closed form expression for such a joint distribution exists. In essence, it allows you to take individually fitted marginal distributions and preserve correlation between those distributions via the covariance matrix. And this is much better suited for single cell RNA-seq data because, as we know, gene expression levels are heavily correlated with each other. So for some subset of non-sparse genes, scDesign-2 will fit a covariance matrix to these. Here's an example of that with a Poisson estimated gene, the LTB gene, or sorry, the SOX4 gene, and a negative binomial fit distribution for the LTB gene. So a more sophisticated class of algorithms is based on diffusion methods. This is a deep learning technique for generating synthetic data, which was originally developed for image generation. The essential premise of these methods is that by viewing data generation as a transition from high entropy noisy data to less noisy synthetic data, you can easily train a neural network to progressively denoise random noise into meaningful structured output. The way this is done mathematically is by generating progressively noisier and noisier versions of training data points and training a neural network to denoise them, to predict the noise that was applied in a given step. And using the so-called reparameterization trick, we can actually compute a gradient on these on the noised data parameter in such a way that a neural network can estimate the noise applied to a given sample. From this, the reverse diffusion process simply becomes a matter of undoing the noising process applied and predicting a probability distribution for the denoised input based on the output. SC diffusion goes further than this by allowing you to condition diffusion generation based on the cell type you want to generate. So they train individual classifiers for each cell type to predict the cell type corresponding to a given noised embedding vector. And based on this, they can add this term, this gradient term, to gradually nudge the direction of the denoising towards a particular cell type. CF diffusion is a similar method, except rather than generating these different classifiers for each cell type, they instead integrate cell type information directly into the diffusion process by embedding each cell type as a matrix Y and using a matrix dot product to use this during training directly, which significantly reduces the training and inference costs. Finally, we have the private PGM method, which Stephen has already discussed, which consists of fitting noisy marginal distributions over single cell expression data, which is binned because it expects discretized inputs, and forming a graphical model among these marginal distributions to generate new differentially private data. So to move on to our evaluation of these various methods, we use the data set provided to us by the challenge organizers, which is the 1K1K data set. This is a data set containing approximately 1.2 million cells sampled from roughly 1,000 donors, contributing between a few hundred and 3,000 or so cells each. And these were split into 14 cell types. You can see here the differentiation tree. I'll mention briefly, and this will have implications later on, that some of these cell types are only very marginally distinct from the others, differentiated over just a small handful of genes, like you can see here in a secondary unsupervised clustering stage used to determine cell types. So here we have the fidelity metrics that the previous presenters have alluded to. I won't go through explaining all of these since the other presenters have already done so, but you can see that while all of the methods do a relatively good job of preserving mean expression levels, this is the correlation coefficients, which are correlation of mean expression levels per gene, and also the maximum mean discrepancy, which measures something similar. Only SC Design 2 does a particularly good job of generating data that clusters together with actual data. So you can see here with the SC Design 2 method, you get a UMAP where the synthetic data is almost indistinguishable from the real data. There are certain bits on the fringes here where you can see real data but not synthetic. But other than this, it's very close to the actual input data. By contrast, with the diffusion-based methods and with private PGM, we see LISIs of 0. So to briefly recap, the LISI is proportional to the probability that a synthetic data point will have a real neighbor or vice versa within its k nearest neighbors. So An LASI of 0 indicates essentially total segregation between real and synthetic data. Now, we're a bit puzzled about why this would happen in the diffusion models. We're still working through this, but we will note that we are not the first to observe this sort of behavior with diffusion models. We did see that in the CF diffusion paper, the authors observed something similar when trying to apply SC diffusion to a spleen-based single-cell data set. And they briefly speculate that this may have to do with having cell types which are distinguished only in a few small genes. But really, this is just a preliminary hypothesis, and we have no strong ideas about why this may be the case. We did successfully manage to replicate their output on the training data sets used in the SC diffusion and CF diffusion papers, which would suggest it's not an issue with configuration or normalization or something like that. But if anyone in the audience has any insights, we would appreciate hearing them. Similarly, for private PGN, we see fairly low quality, which is largely a consequence of the binning, which is far less suitable for the single cell data than it is for the bulk data. So to move on to the evaluation of model privacy, we use the standard attack, the GAN leaks attack provided by the challenge organizers. This is essentially a distance-based attack where you are computing the minimum distance between a particular real data point, which may or may not be a member of the training set, and the various synthetic data points. And based on the minimum distance, you can infer the probability that it is or is not a member. In addition to this, we devised our own distance-based MIA based on the Kullback-Leber divergence between samples. This was targeted specifically at the SC Design 2 model. Because we know exactly how it fits distributions, it's possible for us to fit a marginal distribution using identical methodology over the synthetic data. And by computing per gene, per donor, per cell type, Kullback-Leber divergences, we can essentially compute a score for each donor of the likelihood that they were a part of the training set by summing together these KL divergences. However, when we investigate the efficacy of these attacks, we see that they perform very poorly. Now, really, these last three columns are no surprise. They're performing as well as random guessing because our data set, our membership inference data set consists half of non-members, half of members. And because the synthetic data are so far from the real data, the distance is so large that all training points are predicted as non-members, yielding exactly 50% accuracy. But with SC Design 2, where there is an actual membership inference attack going on, there are certain data points which are being predicted as members and others non-members. We see that it's achieving ARIC that is barely better than random guessing. In this case, actually worse in the top case. And just to drive this home, we do have a graphic here where we show the distance of the Kullback-Leber divergences summed per donor and the proportion of members at each version of the overall score. And we see that there's essentially zero trend here. So this does not seem to be a good metric. So in conclusion, we find that these attacks do not seem to be particularly effective at membership inference attacks with SC Design 2, though of course more sophisticated attacks may yield better results. So finally, I'd like to acknowledge all the people who helped fund this research, most significantly the NAIR project, which gave us the computational resources to do so. And thank you to Sage Bionetworks for their collaboration on this.
Thank you. Any questions from the audience? I have a question. I am curious that when you were showing the results about the membership inference attack, that basically SC Design 2 and then private PGM perform almost similarly, although private PGM is kind of explicitly considering the privacy aspect. So do you have any comments on that part? Or
Yeah.
Whether have you tested more strict epsilon values, or whether it had an impact on the attack performance, basically?
Yeah, so they're performing similarly, but for different reasons. So like we mentioned, private PGM is performing very poorly on this use case, although it performed well for the bulk use case. If you go back to the LISI, you can see there's essentially zero clustering between the synthetic and the real data. This means that the distance between training data points and synthetic data points is so large that essentially every data point considered in the membership inference attack is classified as a non-member, which gives it 50% accuracy. By contrast with SC Design 2, the attack is not performing well, but it is classifying many data points as members. Does that answer the question?
Thank you. Yes, please.
Yeah, thank you for your talk. I guess I have a bit more general question that I guess I'm not very familiar with the data provided, but I'm guessing it's probably some form of like 10x droplet-based sequencing. Do these, or would SC Design 2 generalize if you want to other maybe single cell sequencing types, or would that need to be other considerations that we need to make?
Well, it operates exclusively on normalized counts data. So maybe there are certain characteristics of the distribution that you would need to take into account, but I would imagine as a non-biologist that those wouldn't be too significant.
I guess so too, but thank you.
If there are no questions, thank you again.